Hello, ladies and gentlemen. As you can see, our topic is a study of the mechanism of the drug feeding carrier, using fish as an example. My name is Shen Chen Wang, and she's my partner, Qi Chen. Chinese medicine is a multi-component complex system, and it's hard to screen its efficacy in cell models. At the same time, experimentation animals such as mice, pigs, rats, etc. has long feeding cycle, long times to create animal models with the disease, and high costs. All of the above-mentioned factors make it difficult to carry out large-scale screening of TCM. This is why we selected zebrafish with hepatic cancer as the main reason for the screening of TCM in treating hepatic cancer. At present, there are two main ways to administer drugs in zebrafish, namely direct immersion and intraperitoneal injection. The main disadvantage of direct immersion is the difficult to measure drug absorption rate. Also, it is not applicable to water-insoluble drugs, while intraperitoneal injection may injure the fish, and it's hard to put into practice. Not to mention the fact that there are restrictions on the amount of pharmaceutical usage. Therefore, this study aims at developing a new system platform for the oral administration of TCM in zebrafish to ultimately accelerate the TCM screening process in zebrafish animal models with particular diseases. Zebrafish has a genetic similarity with humans up to 87%, and they reach sexual maturity in a short period of time. Thus, variants can be generated by mechanical or chemical means for research purposes. Therefore, zebrafish have been widely used to study vertebrate embryonic development, disease research, drug screening, and toxicology testing due to its photoperiod-induced spawning and its external fertilization, and also due to the fact that the number of eggs discharged at a time is high. Their embryonic development is quite fast. Furthermore, the eggs and the embryos are transparent. Zebrafish are exposed to light for 14 hours, and the water temperature is controlled at 26 to 29 degrees Celsius. They are daily fed twice. We fed at 9 a.m. and 5 p.m. Every time they are fed so that they can finish it in approximately 5 minutes. Okay, let's move on to the research purpose. First, testing the optimal condition for alginate micropellets coated with TCM and the palatability of zebrafish. Second, to develop a new system platform for the oral administration of TCM in zebrafish. As the picture you see, it is our experimental protocol. First, we prepare the blank alginate micropellets to understand under what condition we could obtain the roundest and the smoothest dry blank alginate coated micropellets with the best vulnerability. Next, we prepare alginate micropellets coated with different feeds and TCM to find out the palatability of medications in zebrafish. Then we determine the rates of encapsulation efficiency, expansion and release, and drying rate. We determine the encapsulation efficiency to find out how much medicine is in the micropellets. As for the expansion, we can avoid zebrafish die after they eat the alginate micropellets coated with TCM. And we determine the rate of release of alginate micropellets coated with TCM to realize the release rate in zebrafish. Table 1 shows the size of blank alginate micropellets after drying for different hours in different concentration of alginate and calcium chloride. As for the concentration of calcium chloride increases, the size of blank micropellets get bigger. We also find out that when the alginate is 1.5% and the calcium chloride is 2.5%, the alginate micropellets after drying 24 hours at a temperature of 40 degrees Celsius can be said to have reached the optimal formability, which is about 10 mm per 10 pellets. Table 2 shows the size of alginate micropellets coated with different feeds at different calcium chloride concentration. At an alginate concentration of 1.5%, irrespective of whether the calcium concentration is 1.5% or 2.5%, The algae and micropellets after drying. The algae and micropellets coated with brine shrimp eggs are relatively bigger. But also after formation and after a drying period of 24 hours or 48 hours, the algae and micropellets coated with feed or with brine shrimps all seem to have reached quite an appropriate size. As for the zebrafish palatability test at a calcium chloride concentration of 1.5%, the algae and micropellets coated with feed or with brine shrimps after being dried for 24 hours have the best palatability percentages, respectively 35% and 70%. The palatability of what is surprising is that no zebrafish which were fed pellets coated with brine shrimps presented a mortality rate of 40%.
这是5分50秒。
Due to the mortality rate, we hypothesized that this was maybe because the alginate micropellets expansion rate. Table 3 shows the expansion rate and the encapsulation efficiency of alginate micropellets under different calcium chloride and alginate concentration, coated with 0.5 gram or 1 gram of chanchong, immersed in water or in intestinal fluid. We can find out that when the alginate concentration is 1% and the calcium chloride concentration 1.5%, no matter coated with 0.5 or 1 gram of chanchong, the alginate micropellets immersed in intestinal fluid would dissolve. Furthermore, the encapsulation efficiency is relatively high. To know the encapsulation efficiency, we have to apply HPLC analysis. Figure A shows the HPLC chromatogram of the TCM standard, ferulic acid, and Figure B shows the HPLC chromatogram of the TCM extract, chanchong. In Figure A, we can see a peak at about 15 minutes, and in Figure B, we can see a peak at the approximate time. By doing so, we can confirm whether the TCM is successfully coated in the micropellets or not. To increase the micropellets' palatability and decrease the mortality rate of zebrafish due to overexpansion of the pellets in the fish, we coated the TCM with 1% of alginate and 1.5% of calcium chloride, and measure the weight and the size of the micropellets every half hour to find the optimal drying time. It can be found that the weight and the size of TCM-coated micropellets stays stable after 1.5 hours. Then we observed the morphological variation in TCM-coated alginate micropellets placed in different fluids every half hour. As shown in Table 5, TCM-coated micropellets placed in the intestinal fluid began to soften in 0.5 hour, and then would dissolve at about 1 hour. In contrast, the micropellets in water would not dissolve would not lose structure until 24 hours. To ensure size consistency across all alginate micropellets, a drying period of 2.5 hours is necessary. Therefore, we use TCM-coated alginate micropellets that had been dried for 2.5 hours for the palatability test. Results show 100% palatability of TCM-coated alginate micropellets dried for 2.5 hours. We also observed the digestion of TCM-coated alginate micropellets in zebrafish at various time points. The experimental results indicate that the micropellets were fully digested in about 6 hours and would not cause fatal abdominal distension.
9分半。